Hello everyone, welcome to the webinar today to learn about how can you use stable cell lines for your drug discovery project. My name is Vivian Zhang. I'm the product manager of cell biology at Genscript. In the webinar today, I'll go through the contents here with you. First, I'll introduce to you about our product history, and then I'll introduce to you the workflow of how we generate stable cell lines at Genscript. And then I'll introduce to you our portfolio, and then point out the product highlights. At the last, we'll go through some case studies. Founded in 2002 and headquartered in New Jersey, USA, Genscript is now a leading company in biotech, providing a variety of products and services in the field of molecular biology, proteins, peptides, antibodies, and cellular analysis. In 2009, we started to provide stable cell lines for Gpcr functional assay. 7 years later, in 2016, we extended our portfolio to also provide immune checkpoint stable cell lines for binding assays as well as for functional assays. 1 year later, we further extended our portfolio to also provide the Fc receptor stable cell lines for antibody binding. Last year, in 2020, right after the COVID-19 pandemic, we have also developed related cell lines overexpressing ACE2 or SARS-CoV-2 spike protein for investigation of the virus binding. So far, we have already more than 10 years of experience in generating stable cell lines for drug discovery, and we have successfully provided more than 200 products covering hot targets in drug discovery. First of all, let me introduce to you the workflow of how we generate stable cell lines at Genscript. We start with constructing the plasmid, including both the target gene as well as the selective gene. The plasmid with the target gene will be selected by the antibiotic selection, and only the target expression plasmid will be used for transfection. And from here, a cell pool is generated with heterogeneous expression of the target gene. Then we perform the dilution cloning to get a single clone stable cell line. And then each clone is further expanded in cell culture, and the one with the best performance is selected as our product. The product clone is then banked in our inventory, further expanded in harvest, and a portion is used for QA before the cell line product becomes deliverables. The first question in recombined stable cell line generation is which host cell to choose. We offer our cell lines in various of host cells, including Troca1, HEK293, HT1080, and Gkit. The host cell of each cell line product is chosen for the best performance of the target gene expression, and we offer as many options as possible. Troca1 is the most popular host cell used in recombined cell line for drug discovery. It is an epithelial cell line derived from the ovary of Chinese hamster. The Troca1 cell we used at Genscript for stable cell line generation is a DHFR sufficient strain. These cells are easy to culture, they are tightly adherent to the vessel, and cost very low to grow with simple medium. Another option we offer here is HEK293. These cells are derived from human embryonic kidney cells. They are widely used due to their reliable growth as well as the propensity of transfection. These cells are compatible for different platforms, especially human disease target. A feature of these cells is that they are easy to detach, so mostly they are grown in suspension cell culture. HT1080 is another option we provide. They are delivered from the human fibrosarcoma biopsy. This cell is a suitable model for NF-kappa B signaling pathway, which is necessary for immune responses. Therefore, we chose this cell line for generating our immune checkpoint reporter cell lines. Jerkit is also our option. This cell is a cell line of human T cells. Therefore, they're the perfect model for in vitro assays to mimic the real immune responses. Talking about the expression of target genes, there are several efforts we have done to ensure the best performance. First of all, there is no tag for the expressed target gene in our stable cell lines. Recent research has found that using the label may cause artifacts in drug discovery results due to the label interference. Therefore, the non-tag technology allows for label-free investigation and higher accuracy. In addition, codon optimization is also performed in our stable cell line, when necessary to increase gene expression level in certain host cell lines. This is due to the nature of different host cell lines. It can be challenging for some genes to achieve high expression level in certain host cell line. Codon optimization can solve this problem without altering the amino acid sequence of the target gene. To select the plasmid with target gene expression, antibiotic selection system was applied. When constructing the plasmid, both the target gene as well as the antibiotic resistant gene was constructed to be driven by the same promoter. A plasmid expressing the target gene can then be selected by the expression of the antibiotics. A variety of antibiotic-resistant selectable markers are used in different products, including hygromycin B resistant gene, pyrimycin resistant gene, zeolcin resistant gene, as well as geneticin resistant gene. A two-step screening method is applied for choosing the product clone at Genscript. The first step is a single clone screening. As you can see in the diagram below, due to the different transduction level and the gene insertion site in the chromosomes, the transduced cells generated from the target gene expression plasmid are actually polyclonal and presented as a heterogeneous mixture. Different cell clones, as shown in the yellow and green, may have different expansion rate. Therefore, in a cell pool after a few passages, you'll start to see the expression level of the target gene start to shift due to the outgrowth of one clone versus another clone. Only when you have a single clone stable cell line, you can have the expression level stable after passages. Dilution cloning technology is applied to generate single clone. In this procedure, the polyclonal cell pool after transduction is diluted to a certain concentration before plating to a 96 well plate, so each well contains no more than one cell. From there, a single clone cell line will be obtained in individual well after several days of expansion. And then, all the single clones are further screened to choose the product clone based on the performance. Highly producing clones are actually rare due to the reason that active regions supporting high gene expression in the chromosome is actually rare. And also, in the case of immune checkpoint stable cell lines, some immune checkpoint proteins can actually induce apoptosis. Therefore, it is very necessary to screen for the clone with the best performance as a product clone. The screening method we use includes both the FACS assays to check the expression as well as functional assays. So, when do you need to use the stable cell lines? To answer this question, we need to take a look into the procedure of drug discovery as shown in the diagram here. As you can see, it is a very long procedure involving many steps. The whole procedure can be broken down into three major stages. Shown in the blue is the drug discovery stage, including target discovery, validation, assay development, screening for hits, leads identification, and optimization. After that is the drug development stage, which also includes filing of the investigational new drug application, or called IND. After that, if you get good results from the clinical trial, you can then move on to new drug application as well as the drug commercialization. In the drug discovery stage, shown in the blue, after a target for a certain disease is validated and the related assays are developed, you start to screen a whole list of drug candidates to identify a few ones with good binding affinity, which are called hits. For this procedure, overexpressing cell lines are needed. Then the identified hits will be carried over to the next step for lead identification, which are the one or a couple drug candidates that can actually active or inhibit the drug target. And this is where the reporter cell lines come into place. Next, let me introduce to you the stable cell line products that we have at Genscript that can facilitate your research, including Gpcr stable cell lines, immune checkpoint stable cell lines, Fc receptor stable cell lines, as well as COVID-19 related cell lines. First, let's talk about Gpcr stable cell lines. Gpcr is also called 7-pass transmembrane domain receptors, or G-protein-linked receptors. They are a large group of cell surface receptors that can detect a molecule outside of the cell and activate cellular responses. The ligand that bind and activate these receptors include light-sensitive compounds, odors, pheromones, hormones, and neurotransmitters, and vary in size from small molecule to peptide to large proteins. When a ligand binds to the Gpcr, it causes a conformational change in the Gpcr, which allows it to act as a guanine nucleotide exchange factor. The Gpcr can then activate an associated G protein by exchanging the GDP bind to the G protein for GTP. The G protein's alpha subunit, together with the bound GTP, can then dissociate from the beta and gamma subunits to further affect intracellular signal proteins or target functional proteins directly depending on the alpha subunit type. Gpcrs are involved in many diseases, including mental, metabolic, immunological, for example, viral infection, cardiovascular, inflammatory, sensory disorder, as well as cancer. As you can see, they are very important drug targets. 34% of all FDA-approved drugs actually target 108 members of this family. 66 potential novel Gpcr targets entered investigation in clinical trials. And there are still 224 non-olfactory Gpcrs that have not yet been explored in clinical trials, and they still have broad untapped therapeutic potential. At Genscript, we offer more than 150 stable cell lines with targeted Gpcr overexpression. This covers 75% of all the potential clinical targets. All the Gpcr expressed in our stable cell lines are human proteins, and we offer a variety of whole cells, including Troca1, HEK293, as well as HEK293T. The Gpcr stable cell lines we offer at Genscript are for functional assays in Gpcr drug lead identification. All the cell lines are fully validated in multiple assay platforms, including calcium flex assay, FRET assay, or the chemiluminescent assay. As you can see here, I'm showing you some example data from our products. In the left panel is a calcium flex assay in the human recombined oxytocin receptor stable cell line. With increased dose of oxytocin, you can see increased level of the retinofluorescent unit change. In the red panel is the representative data from of the CAMP accumulation in the Troca1 GAL15 GLP1R stable cell line. As you can see, with an increased level of GLP-1, you see decreased level of the ratio of 665 versus 620. The next product line I want to introduce to you is the immune checkpoint stable cell lines. Immune checkpoints are regulators of the immune system. These pathways are crucial for the self-tolerance, which can prevent the immune system from attacking cells indiscriminately. However, some cancers can protect themselves from attacking by stimulating immune checkpoint targets. Cancer cells often have tumor antigens on their surface that can be detected by antibodies. Cancer immunotherapy is the artificial stimulation of the immune system to treat cancer, improving on the immune system's natural ability to fight the disease. The Nobel Prize in Physiology or Medicine in 2018 was actually awarded jointly to James P. Allison as well as Takusu Hong for their discovery of the cancer therapy by inhibition of the negative immune regulation. As you can see on the image on the right side, there are activating receptors including CD28, OX40, GITR, CD137, CD27, and the HVEM to activate the T cells. There are also inhibitory receptors expressing on the T cells, including CTLA4, PD1, TIM3, BTLA, VISTA, and LAG3. To target the activating receptors or the inhibitory receptors, agonist antibodies or the blocking antibodies can be developed. We offer more than 50 stable cell lines with overexpression of different immune checkpoints for binding assays. The species of a protein that we express including human, mouse, or monkey. We offer a variety of host cells including CHOK1 or HEK293. The cell lines are for binding assays of overexpressed protein or can be used as immunogen. The high protein expression in the stable cell lines are validated by FACS assays. As you can see here, I'm showing you an example of the FACS assay analysis of the PDL1 expression in our CHOK1 PDL1 cell. Shown in the blue is the expression of the PDL1 in the stable cell line. Shown in the red, you can see that there's little expression of the PDL1 in the control cell line, which is just the CHOK1. We also have 7 reporter cell lines of immune checkpoints for functional assays. They are all human immune checkpoint proteins, and we offer host cells include HT1080, JERKIT, or CHOK1. These cells are for functional assays with a reporter expression. The assay results are either endogenous IL-8 or IL-2. On the right, you can see the reporter IL-8 production assay in the HT1080 OX40 cell line. In this case, the IL-8 is the assay readout. When the cell line overexpresses OX40, it's dosed with different levels of the antibodies for the OX40. You can see increased expression of the IL-8 that is released into the cell culture medium, indicating the activation of the OX40 receptor. The next category I want to introduce to you is the Fc receptor stable cell lines. As we all know, pathogens that invaded into human body can be recognized by antibody through the FAB region. The Fc region of the antibody can be recognized and bound by a Fc receptor. Different Fc receptors are expressed on the surface of different types of immune cells. Therefore, the recognition and binding of different Fc receptor to the Fc region can recruit different type of immune cells and therefore activate the immune response to kill the pathogen. In human body, there are several different types of Fc receptors expressed on different type of immune cells. These include Fc receptor 1, receptor 2A, 2B, 2C, 3A, 3B, and neonatal. As you can see, their expression patterns are all different in variety of different immune sites, and they can recruit the different type of immune cells to the pathogen and activate immune response. The Fc receptor affinity analysis of antibody drug provides information on which type of the immune cells are involved in the immune responses triggered by the drug candidate. At Genscript, we provide 8 most popular Fc receptor members in our stable cell lines, including polymorphic versions. All the expressed Fc receptors are human proteins. They are all expressed in the whole cell of the Troca1, and they are great tools to study the antibody binding, affinity, and half-time test. The last category I want to introduce to you is the COVID-related stable cell lines. In last year, we all faced the global threat of COVID-19. So far, it has been more than 100 million infections and more than 2 million deaths globally, and all these numbers are still rising rapidly. Cell entry of the coronavirus depends on the spike protein binding to a cellular receptor and priming by a cellular protease. Recent studies demonstrate that the angiotensin converting enzyme 2, or called ACE2, serves as a cell surface receptor to bind S protein in the SARS-CoV-2 and facilitate its entry of those coronavirus into the cell. ACE2 has therefore been proposed as a potential therapeutic target for COVID-19. At Genscript, we have developed stable cell lines with high expression of human ACE2 and 2 options of whole cells, HEK293 or Troca1, as powerful cell line tools for drug discovery to treat COVID-19. Both cell lines are validated by the ACE2 expression by its binding to the S protein RBD domain. As you can see in the FACS assay here, the HEK293 ACE2 cell line was split into 2 groups. One group was stained with the SRBD protein with a His tag and then followed by the anti-His fixie. Another group was now stained by the SRBD His and only stained by the anti-His fixie. And then the level of the Hixie was detected by the flow cytometry. As you can see, a significant difference was seen between the 2 groups, indicating the high binding of the SRBD domain to the ACE2 protein on the cell surface. There are many applications of this ACE2 cell lines. One of the applications is screening for the anti-ACE2 antibody. The representative data I'm showing you here is a binding assay of the S protein RBD to the ACE2 on the cell surface. As you can see, with increased level of the SRBD treated with the cell, increased level of the fluorescent intensity can be detected on the cell surface by flow cytometry. Another application of the cell line is to screen for the RBD binding inhibitors. Here I'm showing you an example data of the neutralization assay of SRBD protein by ACE2 protein. The HEK293 ACE2 cell line was treated with 1 microgram per ml SRBD in the cell culture and ACE2 protein at different concentration. And then the neutralization of the RBD was calculated by the amount of the RBD bind to the HGK cells by FACS assay. As you can see, with increased level of the ACE2 added into the cell culture, increased level of neutralization can be observed. In addition, we have also developed SARS-CoV-2 spike protein stable cell lines for the investigation of COVID-19. We also offer two types of host cells, HEK293 and Troca1. All the cell lines are validated for the SARS-CoV-2 spike protein expression by the human ACE2 binding. And the cell line can be used as an in vitro binding assay or used as immunogen. Here I'm showing you a representative data of the flow cytometry analysis of the spike protein expression on the Troca1 spike cells while binding with the ACE2 protein. As you can see in the red, the Troca1 SARS-CoV-2 spike protein cell line was detected with high level of ACE2 binding compared to the control cell line, which is just the Troca1. The next question is, why do you choose stable cell lines from Genscript for your drug discovery project? Our stable cell line products are great cellular tools for drug discovery and are advanced compared to other products by the three key features here. They're stable in passages, they provide high signal versus background ratio, and also they're ready to use. First of all, I want to talk about the stability. When you use cell lines in your drug discovery project, I encourage you to think about your data reproducibility. This can only be guaranteed by a highly stable cell line. Therefore, all of our stable cell lines are tested for their stability for more than 15 passages. Here I'm showing you two example data. In the left is a stability test of the Troca1 MT1 GEF15 cell line. The intracellular calcium mobilization was tested in this cell line for more than 15 passages. On the right side is a stability test of the HT1080 OX40 reporter cell line. In this cell line, the OX40 ligand was added to the cell culture, and the secretion of the L8 was detected in the supernatant. The secretion of the reporter IL-8 was detected for 20 passages and still shown to be stable. The next critical feature you need to consider when choosing stable cell line for drug discovery is how good is the signal versus background ratio. In order to get significant and interpretable data, it is obvious that you need your cellular analysis tool to deliver high signal and background noise as low as possible. One way to examine the S versus B ratio is to compare the signal from the particular cell line of interest to its control cell line. As you can see in the example I present to you here, flow cytometry analysis of the target gene PD1 expression in the PD1 stable cell line compared to its control from either Genscript or the competitor. In the left panel, you can see the expression level of the PD1 as shown in the blue is significantly higher than in its control cell, which is just a Troca1. However, in the right panel, you can see the signal from the target expression cell line is actually overlapping in the histogram compared to its control cell line, indicating this cell line is probably not a single clone and it's not delivering the high signal back versus background ratio. This could be a very serious issue when you use a cell line for your drug discovery research. The next product feature we offer from our stable cell line, which will be very valuable to you, is that they are ready to use. As you may know, cell line generation is a very time-consuming work, but drug discovery is very competitive, especially time-wise. As you can see in the timeline of the cell line development in the diagram here, I'm showing you the number of weeks for each step. In week 1, you need to do your analysis and verification of the DNA. After that, it takes about a couple weeks for the construction of the vectors. Starting in week 5, which is your second month, you can do transfection and selection of the initial stable producing pools. And then it will be another couple weeks for dilution cloning. Only starting in the third month, which is week 9 to week 12, you can do expansion. After that, you still need to do evaluation and choose the top clones. To get the result, you still need to do some QA and also harvest your cells, which takes another couple weeks. And after that, you can get to the final deliverables. The whole procedure takes about 20 weeks, which is 5 months of your valuable time. So why do you waste your time? Speed up your drug discovery by taking advantage of the ready-to-use cell line products. At last, I want to share with you a case study of the Troca oxytocin stable cell lines, published by Professor Pierce with her colleagues from University of Nebraska. In mammals, the non-applied oxytocin structure is highly conserved with the leucine in the 8th position. However, in marmosets, a non-synonymous nucleotide substitution in the oxytocin gene codes for the proline in the 8th residue position. Therefore, in this research, these two versions of the oxytocin peptide were studied for their affinity of binding to the oxytocin receptor as well as activating it by using our Troca oxytocin stable cell line. As you can see in the top panel here, the two oxytocin ligands show similar potencies and efficacies in increasing intracellular calcium concentration in the Troca oxytocin stable cell line. In the lower panel, you can see the Leo8 oxytocin actually shows substantially greater potency compared to the Pro8 oxytocin in activating the potassium channel measured by the membrane potential sensitivity dye, FMP blue. By now, you see why the stable cell lines from Genscript can serve as great cellular analysis tools in your drug discovery project. We have 11 years of experience in generating stable cell lines for drug discovery. We have successfully developed more than 200 stable cell lines expressing various hot drug targets. All of stable cell lines are single clone. They are validated for high expression or functionality. They are stable for passages and present high signal versus background ratio, and also they are ready to use. These cell lines can speed up your project and enable highly reproducible data. You can find more information of the stable cell line products by visiting our website and go to the catalog product sections. If you have any question regarding the product, just go to the document tabs and open the FAQs. Make sure you click on the catalog product section and you will be able to find your question there. Thank you all for joining the webinar today. I hope our products serve you well in your drug discovery research, and please don't hesitate to contact Genscript if you have any question.